Good afternoon. I'm Richard Kincaid, and I'm the CFO of Healios. I'd like to start by thanking the Alliance for Regenerative Medicine for this opportunity, for having us back this year. I feel honored and privileged to be able to present to you today. Thank you so much. And so, for those of you who know Healios, you'll be aware that we have a broad platform with multiple products that we're developing. But today, in the 15 minutes that we have, I want to focus on one of our programs, which is our engineered natural killer cell program. We call these our ENK cells, and we're developing them primarily for solid cancers. So we're a listed company in Japan. I need to show you our disclaimer. Please have a look. Thank you very much. And so the vision for this program is to contribute to the eradication of solid tumors and other cancers by leveraging our unparalleled iPS cell expertise and by augmenting the innate cancer-killing ability of natural killer cells. And so why us? Well, we've been at this for a long time, over a decade. We were founded in 2011 by Dr. Hardy Kagimoto, who's sitting right here. He listed the business on the Tokyo Stock Exchange in 2015, and we've been focusing on developing engineered iPS cell-derived therapeutics. And we're pioneers in cell therapy. Our RPE cells for age-related macular degeneration were the first iPS cell-derived product used in humans in the world. This is back in 2013 and 2014. And since then, we've been developing technologies, know-how, and team that puts us in a strong position to build high-quality iPS cell-derived therapies. We're doing this in Tokyo and in Kobe, Japan, but we're building ourselves up actually here in California and we're excited about partnering with local companies and organizations like CIRM to bring our technologies forward as fast as possible for the US patient population. We have over 100 people across our organization. And we have some key strengths that are very supportive of the NK cell program that we're developing. At our Kobe Research Institute, we have a strong scientific team that's been developing innovative iPS cell-derived therapies and platform technologies. And this team is the one that's created this proprietary best-in-class engineered NK cell platform that I'm going to be describing further today. This team also established a clinical-grade hypoimmune universal donor cell line 2 years ago. It was one of the first of its kind in the world. So we have innovative R&D expertise in the firm. Equally, we've been very focused on getting very good at manufacturing cell therapies. So for this program, we have an in-house GMP facility. We've installed in it an automated 3D bioreactor system. We're already able to efficiently make these cells to supply global clinical trials. So we have robust CMC expertise and infrastructure. We also have clinical development capabilities. We finished 2 studies last year, including the largest cell therapy study ever run in Japan. I want to give you some high-level highlights of the program before we get into the weeds. Our background as leaders in the iPS cell space has enabled the production of a highly potent, functionally enhanced natural killer cell platform. Right? And these ENK cells that we make, they're superior to competitor programs. We believe they are because they're enhanced for recognition, cytotoxicity, and persistence, but also uniquely for migration to the tumor site and for recruitment of host immune cells. And we have in vitro and in vivo data demonstrating robust cytotoxic activity across a range of solid cancers, and we're focused on lung and liver cancer. We're developing next-generation CAR ENK products to target certain cancers where that's required or desired. And as I've said, we've established a manufacturing platform. We're already ready to supply global clinical trials. And in terms of team, we have an experienced international leadership team and over 65 people dedicated to this program. In terms of development, in Japan, the pre-IND process is a multi-meeting process. We're going through that now. We're working towards an IND in 2024. We're also increasing our activity here in the US to get to IND as fast as possible here in the United States. So to drive the point home, how is this different from competitor programs? Right? We enhance for recognition, cytotoxicity, and persistence. Others do that. But we also enhance for recruitment of host immune cells to bring cytotoxic T lymphocytes and dendritic cells to the fight. We enhance for migration, so we get these NK cells to the tumor site and get them infiltrating the tumor. And now there are 4 platform components that I want to highlight. Right? We start with a line, one of our GMP-grade proprietary iPS cell lines, and we edit it. We're editing it so that we get the functional enhancements that we want ultimately in the NK cells that we're differentiating it to. In this engineered iPS cell line, we use it to make a clonal master cell bank, and then we can make an unlimited number of NK cell doses. And then we have a differentiation process, and using that differentiation process, we make very pure and potent engineered NK cells with all the functional enhancements that we're targeting. And this is a product, and we're taking it forward as such as a monotherapy in combination with other drugs. But it's also a platform. We're adding CARs to this to target certain cancers, as I've mentioned. And then we have manufacturing. It's really important, and I'll go through it in a bit of detail shortly. So to zoom in on the cell itself, now our patent's not public yet, and so we haven't disclosed all the edits, but I'll go through them in general terms. Right, a couple of them we've disclosed, the ones that are more common. So we had CD16 for enhanced ADCC. We had IL-15 for improved persistence and survival. And I'll just mention here, we haven't disclosed the data yet, but our survival data is very good. We're very excited about it. And so if there are concerns in the industry about NK cells and the durability of effect or duration of effect, we're very encouraged by the data we're seeing. We had a receptor for enhanced recognition and cytotoxicity, a receptor for improved migration to solid tumors, and we make an edit so that a chemokine is released to attract host immune cells. And collectively, this comprehensive set of functional enhancements, it positions this product to have high potency, but also importantly, broad applicability across a range of solid tumors. Now, this was built on a foundation of highly pure and potent first-generation iPS cell NK cells that we made, right? No edits. Right? We made a differentiation process. We made iPS cell spheres and took them into HPCs and then into NKs, and we expanded them out. We found that our line really liked becoming NK cells, and those NK cells had high potency, they were pure, and they were good at killing solid cancers. Right? And so there's some cytotoxicity data here for A549 non-small cell lung carcinoma, and then they were well characterized as NK cells. I'm going to show you a video, and I don't know if this is going to work well. I haven't done this with a clicker, but this is one of those first-generation iNK cells going up one-on-one against non-small cell lung carcinoma cells. So the white blob you can see that's kind of towards the middle, sitting on a dark sphere. Let's watch what this iNK cell does. It doesn't have any enhancements. So you see it bouncing around. It looks like a bug. It's attacking that cancer cell. And it kills the cancer. And then it moves on to the next cancer cell. So that's what these do, right? So these iNK cells, they're good at killing cancer. And that in and of itself is remarkable. But I want to show you what eNK cells do, or engineered NK cells. So here I have two videos. Left-hand side is the iNK cells, first generation, going against, this time, same cancer, but in spheroid form. It's a mass of cancer cells, right? And on the right-hand side, I have eNK cells against the same type of spheroid. Nothing else in the culture. Starting point is 24 hours, so a day has passed. You can see you already have a ton of bright green on the right-hand side. That's apoptosis. A lot of these cancer cells are dead already. Right? They didn't start that way, I promise that. But let me play the videos simultaneously, and then let's see what happens. So on the right-hand side, you can see all that cancer is dead. eNK cells killed it much faster. iNK cells are good at killing cancer. They're catching up, but it's taking longer. And so you get faster killing, fuller killing with these eNK cells. Again, there's nothing else in this culture. So now I've got two more videos to show you. This is eNK cells versus eNK cells with an anti-EGFR antibody present. Same type of A549 lung cancer in spheroid. Left-hand side, eNK only, right-hand side with the antibody. I'd like to play these separately. I don't know if it's going to work. Let me give it a shot. Okay, the left-hand side it's going. You can see, similar to the last video, eNK cells killing this lung cancer. Does a good job. It kills from the outside. They get inside. They kill from the inside. Turns bright green. These cancer cells are dead. Now, on the right-hand side, let's see what happens. I didn't press fast forward. Right? About half the time, you get the same amount of killing. You get to the end of the video, 86 hours, cancer spheroid has been completely destroyed. And that's the power of enhanced ADCC in our eNK cells. That's one of the enhancements, right? And I haven't put data in here for all the enhancements, but I can tell you we're very excited about how the set of enhancements, this comprehensive set of enhancements, can work together to eradicate, to give us a shot at eradicating these difficult-to-treat solid tumors. So that's in vitro data. Now in vivo data, this is just a couple different lung cancer cell lines. You can see dose-dependent response resulting from eNK cells standalone. It's two different human lung cancers in mice, and you see a complete response when you get to the higher doses. This is a preliminary unoptimized model in liver cancer. Again, hepatocellular carcinoma, human cancer in mice. Again, we see dose-dependent response and good suppression of these liver tumors. We have a lot more animal data that we haven't disclosed yet. We're a public company, and we're working towards a scientific conference in the first part of next year and a publication. So that's got us holding data back, but we're very encouraged by the body of animal data that we've been able to build with these cells. So now I want to go from data to manufacturing, right? As we all know, manufacturing is one of the biggest challenges in our industry. But at Healios, it's one of our greatest strengths. Again, we've been at this, developing products from iPS cells for a decade. And we've already got fully defined upstream and downstream processes established for these ENK cells. Right? The system we use is an advanced 3D perfusion bioreactor system with automated media exchange. We've built proprietary components and controls into it to optimize for quality of cells, consistency, and number of cells per unit of volume. It's a small system. It's 2 by 3 liters. It doesn't take up that much space. But using this small system, 3-liter reactors, 2 of them going together, we make 100 billion cells per batch, right, of high-quality, pure ENK cells. You can see the granzyme being perforin expression levels down at the bottom right, nearly 100%. That's our upstream process. It's very important. But our downstream process is equally important. Right? Why is that? Well, our goal with this program is to make very potent NK cells that kill tough-to-treat cancers. Right? So it needs to be potent. It needs to be safe. Right? But it also needs to be easy to make, easy to administer, and available for everybody. Right? So it needs to be off the shelf. Right? A lot of cancer treatments out there, and they're not all available for everybody. There are patients that are waiting. And so this was painstaking work, and we have a number of patents that are pending for this process. One of them is about our cryopreservation process, where we used AI to design a cryopreservation process. Right? And what were we trying to prove to ourselves? We wanted to make a product that we could freeze, thaw, and it maintains robust cytotoxicity post-thaw. And that's exactly what we've proven to ourselves. Now, to summarize the manufacturing platform, we have a GMP facility and it's operational. We've got an automated feeder-free 3D perfusion bioreactor system installed in it. It produces 100 billion cells per batch at a 2 by 3 liter scale. That's sufficient for clinical trials that we're going to run globally. Right? But we can scale this up and we can scale it out as necessary. We've got proprietary downstream process, including cryopreservation, with demonstrated post-thaw maintenance of cytotoxicity of these cells. And just a quick word on team. We were founded by Hardy Hagimoto, who's sitting in the front row. He is an extremely unique and dynamic entrepreneur and business leader in Japan. He's an MD. To his left is Dr. Toichi Takanaka, the former chairman and CEO of Astellas. He's the guy who made Astellas on merger. He's a revered figure in the pharma industry. Another name to mention, Dr. Glenn Gormley, the former global head of R&D at Daiichi Sankyo, is on our board. A very experienced, seasoned leadership team, pharma executives, biotech experienced folks on both sides of the Pacific Ocean. And we're working together to drive this program forward, not just in Japan, but globally. So now to sum it all up. At Healios, our many years of experience leading in the iPS cell field have yielded a best-in-class NK cell platform. We make unique enhancements to these cells for the most robust cancer killing. Again, we enhance for recognition, cytotoxicity, persistence, but uniquely for migration to the tumor site and recruitment of host immune cells so that we can infiltrate these solid tumors and mount a whole system immune cell attack against them to give us a better shot. We've got promising in vitro and in vivo evidence that demonstrates powerful cancer elimination. We're initially targeting lung and liver cancer, right? But this has broad applicability. We've tested it across a number of solid cancers. We haven't disclosed this yet, but it's worked in everything we've touched. Our proprietary 3D perfusion bioreactor system in an in-house GMP facility it is very important to the program, and it's in-house, right? And we've been at this for a long time. Again, in-house manufacturing means we can control the timing and the quality of the product that we're making. It's so very important for this kind of program. We're advancing towards the clinic in Japan and in the United States. And one last thing I'll mention, I'm probably almost out of time. We're in discussions with a number of potential partners to work together on this program outside of Japan. So if there's anyone in the audience who wants to have a chat, feel free to reach out to myself or Hardy. Thank you so much. It's really an honor. Thank you.